Okay, so I am curious with emotionally stunted emoticon here. Let's say if you expect to see evidence of genetic entropy, say within a biological setting, non-domestication, we would need an experimental organism that has both a short generation time and a relatively high mutation rate, because obviously long-lived organisms such as mammals, a genetic entropy experiment, minimally, it's probably going to have to last hundreds of years, right? Especially if these near neutral mutations are invisible and they have such tiny effects. It's tough to see them in real-time experiments. But let's take something that has both a short generation time and a relatively high mutation rate. So for example, RNA viruses, which I believe are uniquely suited for that purpose. If we could see RNA viruses degenerating quickly based on the fact that they have, both a short generation time and a relatively high mutation rate, would that be convincing evidence to you or no?
RNA virus. So you wanted to see in a natural setting. Okay, so what I have to do is to find a population that is, that has that high mutation rate, not inbred, not domesticated, and test them to see if they do have an influx or of deleterious mutation, if they have a heavy load of
Right.
deleterious.
But I'm just saying, like, if we were to look at something that has short generation time and relatively high mutation rate, that could be, genetic entropy on a small scale versus, say, long-lived mammals on a long scale, right? We can see if it occurs in this type of, like, for example, an RNA virus, they have short generation times and a relatively high mutation rate. So if genetic entropy is true, shouldn't we logically see something like that? Because, for example, bacteria, they have short generation times, but they also have a lower overall mutation rate. But RNA viruses, they have short generation times and a high mutation rate. So the fact that we've seen, let's say, the H1N1 human version, for example, we've seen the H1N1 human version go from a red hot pandemic to a whimper to an extinction event in 90 years. Now, from what I understand, the evolutionists, and I'll get your input here, they don't seem to know why exactly the H1N1 virus went to extinction, but I believe they chalk it up to adaptive evolution. But I don't see how adaptive evolution could cause extinction, because it looks like the virus, okay, and there's a really, really technical paper on it. So I'm just going to kind of go with, in a nutshell, from what I know, it looked like the virus started off more human in its codon usage and ended up randomized. It became worse, okay, at interacting with human DNA over time. And if it is using the wrong codons, that means it will be less efficient at replicating in human hosts. So it looks like there is a decline in functionality, and it matches exactly with genetic entropy. It also explains why the mortality rates went down, and it also helps to explain why the strain went extinct. So is that not genetic entropy in real time? Would you accept that as evidence or no? And if no, why not? Go ahead.
Again, I'll have to investigate the circumstances of what caused it to go extinct.
Okay, good.
Again, I'm not too sure if it's maybe the host environment, the human, if maybe humans found a way to combat the virus and then it went extinct that way.
Well, it
But
looks.
I'm not sure the exact consequences that caused it to go extinct.
Well, it looks like the codon bias declined consistently over time. That wouldn't be consistent with adaptive evolution as far as I'm concerned. And it shows clearly that they got worse, so not better, at interacting with human DNA. Now that correlates to slower replication, and that also means less lethal infection. So the viruses became less lethal because why? Well, they got worse, not better, as I stated, which is exactly what we would predict. At doing their one and only job, which is replicating themselves. So short generation times, high mutation rate. It's been studied, very technical paper, peer-reviewed, I believe, in the NIH journals. I'll send you the paper, okay, because it sounds like you are genuinely interested. I'd like to see your feedback. But, that seems to be genetic entropy in real time. So if all turns out well after you read the paper, would that be sufficient for you to validate the genetic entropy thesis? Go ahead.
Sure. Again, if I do read the paper and I find that it seems that the virus itself was getting weaker instead of the human immune system getting stronger against it, then sure, then it should count as genetic entropy.
Because now there are swine flu versions though now circulating, so you know that.
But that is evolution. Some changes worked, some changes did not.
Yeah, but this went literally in 90 years, Snake, from a red hot pandemic to a whimper to an extinction event. They got short generation times, high mutation rates. So,
but other strains are still extant. They survived. They didn't.
Of course, and I believe they are also now experiencing degeneration as well. So they're on the verge of extinction as well. Just like, because remember, it took 90 years for the H1N1 human version to go extinct. Now, I mean, the paper is really, really technical. It's long. It's peer reviewed too. So, I mean, I'll send that to you guys and see, because obviously if we agree that there's degeneration in domesticated animals and species, inbred. But the thing is, I mean, RNA viruses in general, they are known to be prone to error catastrophe and spontaneous mutational meltdown. I believe that that's pretty well documented. Don't you agree, Snake?
Yeah, some of them.
But the thing is, viruses can also persist for very long periods of time in a dormant state, and then they can re-emerge from natural reservoirs. So, you know, whatever's going on with the swine flu versions or whatnot. But I mean, the H1N1, it did go extinct. That's a fact. You know, read the paper, see what your explanation is to it. And then even
how come this mutation accumulation rate is not uniform in all populations?
Well, it depends on, so for example, RNA viruses, you got, you know, short generation times, high mutation rates. That's why they're a good candidate to look for experimentally for genetic entropy. We got humans, 100 new mutations per person per generation. E. coli bacteria, I believe, is about 1 in 1,000, plus way more rounds of selection.
Yeah, bacteria actually have a really high mutation rate.
No. They have an overall lower mutation rate.
What do you mean by that? Overall lower than what?
Well, they have 1 in 1,000 cell divisions is roughly a mutation in, I believe it's the E. coli. In the paper I have talks about E. coli. That's about 1 in 1,000, which is a fact. I debated Dr. Stefan Frello, and he had that published. Yeah, mutation rate.
Well, the mutation rate in bacteria is about twice that of humans per generation.
No, humans are 100 per person per generation. Bacteria such as E. coli is 1 in 1,000.
What you cited the other day in a video I watched was 1 times 10 to the negative 9th for bacteria per generation, per division, and in humans per generation it's 0.5 times 10 to the negative 9th, which is exactly half.
Well, what I stated was bacteria, they are, well, for one, E. coli, for example, 1,000 times smaller than that of humans. They're also, you know, bacteria are more specialized and perform fewer functions. That means any letter change is more likely to do something that natural selection can see. So that's the whole point. I mean, nobody, you know, genetic entropy doesn't predict that bacteria will actually go extinct first. Because, you know, it's more likely that a small change will produce in a bacteria a large enough effect that it will make a difference in the number of individuals, say, carrying that trait generations later. But I've always said it's about 1 in 1,000. So I'm not too sure where you, I mean, like I said, Dr. Stefan Frello, same thing. You can go look at his debate with me. He had it cited. It's about, you know, it's 1 in 1,000. Matt, I think you have the paper that confirms that. But regardless, I mean, bacteria are more simple organisms, and they have a lower overall mutation rate. Do you disagree with that?
Lower than what? I mean, I just made a video about this, like responding to that video, so that'll be out tomorrow.
Yeah. That's fine. So if you made a video then, so if we actually look at, let's use, are you familiar with Lenski's experiment? Like, what do you know about Lenski experiment, the Lenski E. coli experiment, Snake?
Well, there were populations that improved their overall fitness, and some populations that did not, that went extinct.
Well, in a narrow sense, there were some adaptations observed, but in all 12 in all 12 experimental populations, the functional bacterial genome size has shrunk. Like there's literally less total information. You know, they've actually lost genes for adaptive purposes, but at the end of the day, the net effect in the long run is degeneration. So I understand that according to your philosophy that's still evolution. But
is that in the study that there's degeneration?
Yes. It talks about that
for every single population.
The 12 experimental populations, the functional bacterial genome has shrunk. There's papers documenting the beneficials that were observed. Okay, so yeah, it is documented, and a lot of it was due to loss of biological functions, loss of, well, I mean, reductive evolution is known in bacteria. You can look for papers, reductive evolution in bacteria, and you'll get a ton of papers. Bacteria are just known to do that. You know, they'll lose genes, especially in artificial environments, you know, genes that are not necessary in that environment. They'll adapt by losing genes. It's adaptive optimization. But to be honest with you, it's eventually stasis. I think it's called escape from adaptive conflict. Matt, am I using that term correctly?
Yeah, exactly. That's when a gene usually has about 4 to 5 different functions, but it drops and reduces itself to just one. Yeah, that's kind of what the bacteria did, because now those bacteria in Lenski's experiment, they were only fed one type of food, so they got lazy. They actually don't move, and they can never live in their original environment like the other bacteria can again. All bad for them.
So I know, so Snake, you love, yeah, great points, Matt. You love to go to the fossil record, you know, to validate, you know, universal common ancestry. But if we look at the long-term E. coli experiment, I think it's more than 60,000 generations now. Matt, what is it, about 100,000 generations now?
More, yeah, it's okay. It's way up there. It's approaching almost 2 million years of human evolution.
So what's funny is this famous evolutionary experiment, okay, proves that in deep time, which you're looking to, Snake, when you look to the fossil record, even given a model population that should be optimal for validating evolution, proves that populations don't evolve, and instead they devolve. So I understand that you stand by your philosophy, but based on this real-time observable experiment, it's science fiction. You know, it's
well, it actually proves that they survive a lot longer than they should if there was genetic entropy, and they have evolved new proteins and new functions.
No, they've.
But I don't think this is really that productive, because I can admit like that there is genetic entropy. There might be a time limit on life forms, but that doesn't really comment on evolution. Like I could even give you that the dating in the fossil record is off, but that doesn't really affect the theory of evolution.
See, but the thing is, you know, you said that fitness was increased in this experiment, right? But the thing is, you can even look at the paper, like this fitness increase, it's only in a narrow sense versus a broad sense, because in broad sense, if you're gonna, the best definition of fitness is total functionality. Would you not agree?
That seems kind of like a word salad.
Okay, well, what's your the best definition of fitness should be total functionality, not just looking at a single trait.
Fitness for your environment.
Right. So the fitness for this environment was like if you use the full meaning of fitness, total functionality given this environment, all 12 of the population have experienced a net loss of function. And so they have reduced total functionality as needed in the real world. Okay. Yes, to adapt to this environment, they've lost genes, but bacteria do that in the real world too. So real world
they've gain function in some genes, and a lot of the genes that they've lost function for, I mean, those can be beneficial mutations. There are loss of functions that are beneficial.
Well, yes, according to like your belief system, you know, losing genes and, you know, breaking down pre-existing functional systems is going to take your bacteria-like organism to a biologist, but there's no science to support that. In our new house, we tore down, just one sec, in our house, new house, you know, we're doing renovations, we tore down the wall that separates the kitchen and the living room. That was adaptive. But guess what? We destroyed that wall that separates the two rooms in order to make a bigger room. Now, at the end of the day, if someone told me, how'd you build the house? And I told them that, well, that's not explaining how I build the house. That's just, it's just like these bacteria, you know, it's a net loss of information. You look to a trade-off, of course, but the question is the trade-off sustainable? And obviously it's not, because if fitness is increasing in a very narrow sense, okay, you know, a certain portions of the genome is increasing in fitness, but the greater portion is simply just losing information and degenerating. Well, at the end of the day, if it comes down to a net gain versus a net loss, that's a net loss of fitness. And, you know, you can disagree and say, oh, that's evolution, you know, that's how evolution works, but there's no science to actually back that up. You know, the long-term E. coli experiment, the largest evolutionary experiment there is currently, proves creation and not evolution. Go ahead.
Well, we could be evolving towards these total mutants, and it still wouldn't really comment much on evolution, especially since you don't, I mean, you haven't calculated a time limit on any of these things. And the time limits that we do know of are pretty short.
Matt, wasn't there a time limit, Matt?
Oh, yeah. Yeah.
Dr.
John Sanford predicted the extinction of the human race in 1997. He said at 350 generations. We're at 303 right now.
Okay. Well,
see the thing. Well, I mean that answers your question, right? So the thing is,
that doesn't happen. I got how many generations do we have left? 47.
Yeah.
I guess we'll all be gone by then, unless we all eat fruit.
Oh yeah.
We're all going to go on the raw. We're all going to go on the raw mat diet. Well, I want to say one final thing here. So, at the end of the day, you know, Lenski's team of researchers, you know, they did sequence the genomes of these E. coli, so they were able to track all the mutations that arose in this experiment. Now, when these key mutations were examined, so there were some beneficials, you know, like I said, I've never disagreed with beneficial mutations, but they are environmentally dependent, and it's about context. But it is clear, given the data, that the beneficial mutations, they did involve loss of function. I think one was even based on a disrupted regulation. And it's this long-term E. coli experiment. It's not undergoing rapid evolution there, Snake. It's actually undergoing rapid degeneration. I mean, that's the exact opposite of what you need to take your fish to fishermen, to build a genome. You're not going to build a genome by degrading.
Like synaptic pruning, because children are born with more synapses and more brain cells than adults, and we actually gain more information by losing brain cells.
Wait,
you're saying what's that? Were you going to say something, Matt?
I've actually studied that they've actually found a lot of losing of intelligence and emotional abilities from mutations recently. Matter of fact, the Science Cell Press, the Journal of Genetics, they suggested that we're losing our intellectual and emotional capabilities due to the increased web of genes that are endowed with in the brain for susceptible mutations. Based on their calculations, they even said that the frequency of deleterious mutations in the human genome are said if we have about 120 generations left until we've sustained more harmful mutations. They said that they're linked directly to autism and things like that. I can find that study for you.
And we all become psychopaths or something.
But
there are genes where, like the HIV resistance, for example, the inactivation of that gene confers a resistance, and there's no negative effects that are known. And in whales, the baleen gene is just a part of a normal tooth gene. So you can actually take away information and get a different meaning from it.
Now, what on a genomic level happened in this H1 or the HIV-1 immunity? Was it a loss of a deletion, or like what was it on a genomic level?